Okay, we need to get started. I appreciate everyone taking their seats. It's a real pleasure to see so many people in attendance here. My name is Michael Perella. I'm the chair of the Department of Entomology and Nematology, and we're here really to celebrate the department. One of our tremendous graduate students that has come through our system, and I think we need to be very proud of him and many others that have graduated from our program. I think, as everyone knows, we've changed our name recently, right? We've changed it to the Department of Entomology and Nematology. And so now if we say we're one of the best departments in the country, I think there's only two of us really. So, in that sense, we're either one or two. But, that's beside the point. Even if we were just entomology, we do consider ourselves to be one of the best departments of its kind in the nation. And so, we've talked a lot recently, and the campus has done a lot of evaluations of different departments and trying to use metrics to measure strength. And, we have done very well in all of those. There's no question about it. But I think, we all agree that, a way to measure the success of a department is to look at how well our graduate students do. So in that context, we have some tremendous students now. We've had some tremendous ones in the past. We are honoring one of those individuals today. So I think it's fantastic. Now, the seminar series is named after Tom and Nina Lee. And so I thought, spending a little time, just a little, talking about Tom especially. And Tom Lee was a professor in the Department of Entomology. He's passed away some time ago. He was off campus. He was at the Shafter Research Station down in Southern California. Very famous, applied IPM cotton entomologist, but exceptionally interactive in the department. He came to every department meeting. He had graduate students in the department as well that have gone on to do great things. And, in that context, he recognized that what we needed was a fund, an endowment that would enable us to bring some of our great graduate students back to the department, celebrate their accomplishments, and sort of measure our success through their success. So Tom and Nina Lee have donated funds and have enabled us to do this, basically to honor our past graduates. So we are honoring Dr. Ken Haynes, and Ken is, give a little bit of his background. You can read his biography in the handout that you have, and it's on the web as well. Ken has gone on to do some fantastic things. I think originally from the East Coast, so he graduated from the State University of New York in biology. Then he came out to California, I think in 1976, and he became a graduate student of Martin Birch. Now, Martin passed away a few years ago, so hopefully Ken talks a little bit about Martin. But Martin had a tremendous program in chemical ecology. And so in that context, really, at a time when chemical ecology as a field was really defining itself. And so one of the things that Ken has done with Martin, he published a book on insect pheromones many years ago. And again, that's sort of a landmark publication in that field. And then Ken has gone on to publish two other books on chemical ecology, and both with Jocelyn Miller from UC Riverside. But let me back up and talk a little bit about Ken in the department. So he was a graduate student here, and many good things about his interactions in the department. He was a presidential fellow as a graduate student. He also received an NSF fellowship as a graduate student. He also received I guess I say R because we don't really have it anymore, but basically a sort of an outstanding graduate student award for teaching, and maybe we need to resurrect that. So very interactive as a graduate student, very successful, tremendous number of publications while he was here. And that ended, I guess, for him in 1982. He got his degree. He went down to UC Riverside, where he was a postdoc for a number of years with Tom Baker. And I had the pleasure, I was an assistant professor at that time at UC Riverside, so I interacted with Ken. We actually have a publication together, too, which is really remarkable to be linked to someone like this. And then in 1986, he finished his postdoctoral experience, and he went to the University of Kentucky, where he's had a spectacular career as a chemical ecologist. So, to talk a little bit about his success there, tremendous number of awards. He became a fellow of AAAS a few years ago, more than 100 publications, on a number of editorial boards. Again, I think you can look in the program and see his many successes. He also is the holder of sort of an endowed chair in the Department of Entomology on the University of Kentucky campus. So he's the Bobby C. Pass Professor of Entomology. And for those that don't know Bobby Pass, I think, created the Department of Entomology at the University of Kentucky. He was chair there for a tremendously long period of time. He was president of ESA, really an established figure in entomology, and basically an endowment was created in his name, and Ken is currently the holder of that chair. So Ken has a lot of really broad interests, I think. We're going to hear about some of that today with respect to bed bugs. But I urge you to read a little bit about his biography. As you all know, we are looking for a chemical ecologist now, and we're in the middle of that. I shouldn't say the middle, we're at the beginning of a search. And obviously, Ken's a little too old, I think, to be considered for that position. And I shouldn't say old, I should say experienced, right? And Sharon, where's Sharon? She's probably shaking her head at me. So we don't want to get into age discrimination here. But Ken's very happy in Kentucky. He has no interest in coming back, I don't think, but we'll see. And the more I talk, the more I'm getting into trouble here. So getting back to my original point, we're looking for a chemical ecologist at the assistant professor level. And so in that context, we are interested in someone that's going to be doing chemical ecology, I think, on the scale of what Ken is doing. So that's what I'm trying to say. So we look at Ken and maybe look at his program as sort of a template for what we'd like to hire here. So I think with that, I will turn the podium over to Dr. Haynes. Let's welcome him.
Oh, thank you very much, Michael. I appreciate that introduction, and it's a great pleasure to come back here after 30 years. I've seen a lot of familiar faces and lots of new faces, and had some great conversations today. So it's been a fantastic trip. I've gotten lost on the campus only 3 or 4 times today, so I don't recognize the buildings anymore or where my place was on the campus. But it's starting to feel familiar again, and it's a great place to have worked as a graduate student, and I have only fond memories of this place and the education I got here and the people I worked with while I was here. I'm going to talk about bed bugs, but bed bugs are a really recent adventure for me. I've been pulled into bed bugs by the interest of one of my graduate students about 8 years ago. And I will get to that, introduce that individual in a little bit, but I do want to say a little bit about the person we're honoring here at the top, Tom Lee. And he was someone that I recognized as a gentle giant within the Department of Entomology. And I think he had the respect of all of the faculty and students in the department, and I remember his trips up here. Of course, he was stationed away from campus, but still had an impact on the department. The other two in this picture are Martin Birch with the beard, and he was my major advisor when I was here. My interest then was in pheromones of moths, and he gave me the opportunity to work on moths while I was here, so that was great. He did leave before I finished my degree, and Sean Duffy, who's also passed away now, both Martin and Sean have passed away too early, and Sean Duffy stepped in and helped me finish up my degree, so I'm indebted to both of them for what they've done for me and how they've inspired me over time. Martin Birch was someone I've published with from the graduate school days, but also someone who gave me opportunities to develop in ways that I really appreciate, including joining him on publishing a book on insect pheromones that Michael mentioned. I was really shocked when Sean Duffy passed away, but he had also been an inspiring person in terms of my career development. When I started working at Davis, I started with a cabbage looper moth, which I ended up diverting away from later in my degree program to work on the artichoke plume moth, who's also pictured. Cabbage looper and artichoke plume moth are both pictured in this slide. The artichoke plume moth became my dissertation work. The cabbage looper actually was an insect that I came back to and have worked on and off with, mostly on with for the past 25 years. So I came back to the cabbage looper after I graduated.
Ken, I don't mean to interrupt, but did Harry Lang work with you at all?
Harry Lang, of course, introduced Martin to the artichoke plume moth, and
which is named after Harry as well, the species.
Yes.
Named, right?
To get on to the bed bugs, give you a little bit of background. When I give a talk on bed bugs, at the end of the talk, when I presented lots of research data and information, everyone asked general questions about bed bugs. So what I'm going to try to do is integrate some of the studies that I've conducted with an overview of their biology. And so it'll be a combination of that information about bed bugs in general and some of the research directions that I've taken. My work did not start 20 million years ago, but bed bugs and their relatives probably originated as bat ectoparasites. And so they've been around for quite a while. And they're found in the tombs of the ancient Egyptian pharaohs in fossilized form. So we know they've interacted with people for quite some number of years. I think 300 years ago was probably the peak of integrated pest management when gunpowder was used in bedbug control, as it's illustrated in this slide. Gunpowder is packed around the seams of the sleeping area and ignited. And we've had people who've returned to that in recent years out of frustration. Maybe not gunpowder, but fire. And it's another one of my favorite stories is that urine was used to extract plant allelochemicals that had defensive properties, insecticidal properties. And so the quote for how to use urine with bed bugs is to extract these two poisonous plants with the appropriate amount of urine, no specifics given, and then dose the bed area with that. And I'm not sure whether it worked or just discouraged people from sleeping in them. But I guess it worked if it did that. Our ancestors, in terms of our great-grandparents, controlled bed bugs, which were always a problem, by intensive cleaning efforts. And this involved things like taking the straw from mattresses outside and replacing that, to soaking bedding areas with kerosene, which I've seen in more recent years too, in the last few years as well. And just intense spring cleaning, with the effort being just to knock the populations down temporarily and then repeat the process the next spring cleaning when the bed bugs were bad again. So some of that was in our history, but then insecticides came on the market, and there were some real innovations 80 years ago with insecticides. This product, Bed Bug Murder, was one of those, and it had 100% active ingredient. It was right on the label, 100% active ingredient. That's all the information we have, so it must have been a top secret. 60 years ago, bed bugs started to disappear. And this was when DDT was used extensively for control. People back from that era said bed bugs disappeared from the environment over the years. And there are many entomologists for 3 generations that hadn't seen bed bugs, despite the the commonness of them in the literature. But now we have seen them again. Already when DDT was being used, bed bugs were being reported to have some resistance to DDT as an insecticide. So the writing was on the wall that that would no longer be an effective product. About 15 years ago, bed bugs started showing up again in significant numbers. First in hotels, and I blacked out the name of this hotel, but right now that's immaterial because there's every hotel that I know of would have to be dishonest to say they've never had an encounter with bed bugs, at least on the East Coast, and I suspect the same is true out these ways too. And so now bed bugs are common, not just in urban areas, but scattered. These are reports. There's actually a online site where you can look up where bed bugs have been reported in your town, and that's what made this map up, is accumulation of all those reports. Of course, not all reports are accurate, but many of them are. In terms of the pest management industry, the urban pest control operators view bed bugs as the most difficult urban pests to control in the current environment, far above cockroaches and termites and ants. And that's because there are good solutions available for cockroaches, termites, and ants, at least relative to bed bugs. So that's a recurring theme. They're hard to control. This is the business end of a bed bug, the sheath that contains the piercing parts of the mouth, viewed both from the ventral aspect as well as from the side view. And they're impressive insects. That's a face you would learn to love, I think, over time. One of my colleagues in the department teaches an introductory entomology course in one of our service courses in the department. And for several years, he would put bed bugs on his arm and demonstrate their engorgement over time on bed bugs. And that was until this year. And this year, when he did that experiment, he started for the first time to get a reaction to those bites, which is documented in this series of slides. And he said that demonstration is canceled for the future. And there's probably good reason, because here's an example of a severe reaction to a bed bug bite. This includes the obvious welt, but also some swelling that extends down the wrist, and it's very severe. This is my colleague who at University of Kentucky, who demonstrated this, is 61 years old. But this is a different 61-year-old entomologist who allowed bed bugs to bite him. And some of you from 30 plus years ago might recognize Mark Feldlofer's name. And this is the entomologist. At that time, in Briggs Hall, he and his graduate student comrade there, Mark Eberly, would feed bed bugs on themselves over time. And at that time, he did not have severe reactions. But now, 32 years later, he's getting this kind of reaction. He's also back in bed bug research in a big way. And he's got to learn how to avoid getting these bites. Now, bed bugs are an economic issue because they can be very expensive to control, especially when their populations have established in multi-unit dwellings for a long time, and populations have built up and spread through rooms. There's some examples on here of the kind of expense it could take to do a thorough job on control. But there are other types of expenses. We're in a society now where litigation is more common, and many suits have been filed against hotels related to bed bug bites. And so sometimes these have resulted in large settlements. Hotels can be, brand names can be damaged. If you start to associate bed bug with Hilton, then you start to say, well, maybe I'll stay at the Marriott this year instead. So the hotel industry is very concerned that they not be labeled as bed bug capitals of the hotel business. I think one of the more serious parts of bed bugs in our society is that cost-effective means of controlling bed bugs are often out of the reach of the people who suffer from them the most. And so the lower economic strata have significant problems with bed bugs, not because they're any tastier than anyone else, but because they can't afford to get rid of them in a timely manner. And so their populations build up in these communities, and these communities will serve as reservoirs for everyone in our society. So bed bugs have become an issue along these lines. It's clear that bed bugs are not a pest of that sometimes the cockroaches are associated with filthy environments or something like that. Bed bugs are not that kind of pest. They can establish anywhere, but the issue with respect to economics is that individuals who can't afford it or can't afford to throw away their beds suffer from the consequences of bed bugs more. They're also a psychological issue. And in this context, the suffering is mostly related to a stigma that's associated with bed bugs in a modern society. Unlike our great-grandparents who learned to live with them, in a modern society, they're viewed as a no-no. I've had family members say they don't want their children to come home from college because there are bed bugs in that college. Maybe not even in the room they were staying in, but at that college. So it's a real, poses a psychological problem for many people, and in some cases really leads to insomnia, anxiety, depression, and even cases of post-traumatic stress disorder have been reported. They can have dangerous consequences too. The first case on here, 25 bug bombs, and bug bombs are completely ineffective against bed bugs. 25 bug bombs were set off in this apartment, and on top of everything else that you can imagine be used. And eventually the husband was doing this, and the wife was ill, and she eventually passed away with this kind of exposure to multiple agents being in part responsible. But you can see some of the old techniques coming back as well with the fire, the kerosene, and the internet has allowed markets for insecticides that shouldn't be used in a sleeping environment to come back. Now, there are quite a few hypotheses stated in the literature about why bed bugs have come back, because they disappeared essentially. They're still around in pockets, many places, but most entomologists didn't interact with them. And this is a list of some of those hypotheses. The one that I think plays a major role from some of the work that I've done is the last one on this list, insecticide resistance. And I'll explain a little bit about why I believe it to be the case. With the graduate student I mentioned before, Alvaro Romero, who's now an assistant professor at New Mexico State University, we looked for insecticide resistance in bed bugs around Lexington and Cincinnati, which seems to be a hotspot for bed bugs. And we found resistance. The first population that we looked at intensively had a 10,000-fold resistance to pyrethroid insecticides. So that means you could put in 10,000 times as much pyrethroid as would kill a susceptible strain and not kill them effectively. We couldn't get up to a lethal concentration of deltamethrin with these bed bugs. Since then, we've looked more intensively around the country for bed bugs, mostly concentrated on the East Coast, but one population out here in California, two populations. I'll show you one later. There's the other one. This is where we found susceptible populations. One population from Los Angeles, California was susceptible. There was also one from Los Angeles that was highly resistant to pyrethroid insecticides. And then there was one population in New Jersey that was sort of intermediate level of susceptibility. So where pyrethroid resistance seems to be widespread. When we've looked at the specific mutations related to DDT and pyrethroid resistance, we found that 87% of populations that are shown in these pie charts scattered around the country, 87% of those populations showed one or two KDR mutations. So KDR mutations were widespread. I think one of the interesting things here is that first population that we looked at from Cincinnati, Ohio, did not have KDR resistance. It had a stacked layers of other forms of metabolic resistance, but did not have this target site resistance that's associated with DDT resistance and pyrethroid resistance. So of those 13% that were not resistant, there are some of those that have high levels of resistance. So we don't know what the proportion overall of high levels of resistance is, but it's very high. If you're in the market for a mattress, would you buy one? These are cheap and readily available. We've had a lot of experience going around to apartments in northern Kentucky area and Lexington and further into Ohio. And one thing that's common is in pest control with bed bugs is that they throw mattresses away and throw them out near dumpsters. And before the garbage collector comes, they're gone off in another direction to another home, another happy home, temporarily. So this is one way that insecticide resistance interacts with the sort of the movement of furniture and bedding and so forth. If you finally get frustrated that your control tactics are not working for bed bugs and you throw out your mattress, you're spreading the insecticide resistant bed bugs to other places. One of my grad students went around and looked on our trash day, had mattresses on the curb, and she found three out of three mattresses she found on this one trash day had bed bugs on them with no indication that they were infested. Some of the characteristics of bed bugs. My interest has been in behavior and chemical ecology. So some of the characteristics of bed bugs have really been of interest to me. Bed bugs hide really well, they aggregate, they mate in a very, very unusual way, and I'll come back to illustrating some of these in a moment. They forage for blood at night. They feed on warm-blooded mammals, including us and birds, chickens. They can be pests in poultry houses. Finding host is is influenced both by heat and carbon dioxide. Jim Carey, I saw one of your student videos talked about that point of the stimuli that influence bed bugs. They depend on blood meals for each molt, and one of their principal characteristics is they can survive for months without a blood meal. So it's an adaptation that allows them to survive periods when the host is away. So here they are hiding out in a little hole drilled in a piece of wood. There are actually about 50 bed bugs packed into this 3/8 inch diameter hole. They're stacked up one right behind the other. And they aggregate. The aggregations are pretty impressive when populations get high, but those aggregations have everything in them except the kitchen sink in terms of what's up with the bedbug life cycle. They're nymphs and adults, eggs and feces, and all of those occur around cracks and crevices, seams of the bedding area. Their mating is unusual. On the right, the sickle-shaped part is the intermittent organ of the bed bug. Oh, I'm supposed to warn some people to divert their eyes temporarily. You'll have to do that for the next slide as well. The females have an adaptation, part of their cuticle, to receive the puncture wound. But the males actually puncture the cuticle of the female and deposit sperm to the inside of the female, where the sperm must migrate to the reproductive organs through the hemocoel. So it's an unusual mating behavior. And here it is. I hope you can see that one, but the male. And it took an effort to penetrate the cuticle. So it's a traumatic thing. And if we maintain colonies that start with a 1-to-1 sex ratio of bed bugs, they end up all male after a month or so, because the multiple mating will eventually kill the females off. So we typically maintain our colonies with skewed sex ratios favoring females, so the multiple mating doesn't do them in. This is Alvaro Romero, who I mentioned before. One of the studies he did was on circadian rhythms in bed bugs. And here's a series of photographs of individual bed bugs in these individual wells. This is what it looks like during the daylight shots, where they become less and less active. And then when it switches to the gray, my wife has to tell me the color scheme because I'm red-green colorblind, so I was told that it was green and gray, but I'll have to trust her on that. So when it goes to the gray, it's the nighttime conditions with an infrared camera. And under those conditions, under light conditions, they slow down their movement. Under nighttime conditions, they speed it up. And this illustrates sort of the summary of what he found with their movement patterns. Consistently, they're nighttime foragers. And this is what you'd expect from an insect that would suffer if it were discovered. I think one thing that's interesting, if they are derived from bat bugs, then they must have flipped the life cycle responsiveness that they had somewhere in their evolutionary history. The real demonstration that it's an endogenous circadian rhythm is to shift them into continuous light. And some people have thought that this would prevent bed bug bites if you just left the lights on, but it's only a temporary benefit in discouraging them from foraging, as shown here, where some of the large 5th-inch star nymphs continue to come out every 24-hour period, even when the lights are on continuously for those periods. So they have a clock, internal clock, that keeps them going on this 24-hour cycle. In terms of the blood feeding, I'm not going to show this whole video because it takes a few minutes to show, but this is a tiny first instar nymph about the size of the head of a pin. You can see them, but you have to look very carefully for them. And first instar nymph will increase its body mass about 3-fold while it fills up with blood. So that's what you're watching, but I haven't suffered any consequences of bite reactions yet. Maybe in 32 years. Here I'm just showing a video of a quarter that I put in an oven at 50 degrees centigrade for a while, and then brought it out to that same hole that you saw before with the bed bugs hiding. And they're interested. They're coming out of the hole, and they'll make probes at this quarter that's warmed up to above body temperature. Actually turn away from it when it's too hot, and of course as it cools down, they'll get closer and closer. Some frustration here, I think, as they discover that it's not worth a plug nickel. Now, heat is really what governs their immediate feeding behavior. We can have the early instar nymphs or adults tank up on any kind of food coloring we choose if we heat it to the right body temperature. And I think the applications for this are enormous. I don't know what they are yet, but maybe as Christmas decorations on a small tree. One of my students, Becca Hassel, studied the responses of bed bugs to heat and carbon dioxide in a small arena about the size of a sheet of 8.5 by 11 sheet of paper in the laboratory, where we could well contain the bed bugs while we videotaped their movement patterns. And the source cylinder here is one that we could regulate the heat, the temperature to whatever temperature we wanted. And we could also introduce CO2 in a regulated way where we can control the parts per million of CO2 that we put out. Now, when humans exhale, the parts per million of our breath is about 40,000 parts per million. And that's well above anything that the bed bug is likely to experience. If you get close to our skin, the CO2 concentration is about 800 parts per million, which is probably a more realistic stimulus in terms of what a bed bug is experiencing. And the atmosphere itself has 400 parts per million CO2. So it's a small increase could have an effect, and that's what we were interested in looking at in this study. We used video tracking software to follow the bed bug movements, and this just illustrates a couple of their paths. The top one is a control condition where the source cylinder was not heated and was not releasing CO2, and the bottom trace is where the heat cylinder was put at 40 degrees centigrade, but there was no extra CO2 released. And that heat, 40 degrees centigrade, is about human body temperature. And in terms of the bed bugs that made it to the source, that 40 degrees centigrade had a big effect on improving how many crossed into that area where the cylinder was. You could go to higher temperatures and you could get more of them start to orient in that direction, but many of them start to turn back before they get there. We actually studied the movement directions and when those patterns change. With CO2, I'll just show you a CO2 slide here. With CO2, we couldn't see any change in their movement patterns associated with distance from the source in this relatively small space. With heat, we could see that within 10 centimeters, the direction of their movement started to change. but that's relatively close to the bait. I think what Alvaro was finding with that circadian rhythm study was that a lot of how bedbugs find us is about just moving around. And I think what CO2 does is not so much as an attractant, but it stimulates them to be more aggressive in their moving around. They move more frequently when CO2 is present. But CO2 I don't think has a directional component, with heat playing a role only at very close range. Of course, the thing I was very interested in was exploring aspects of chemical communication in bedbugs. And there is a literature going back to 1974 about bedbug alarm pheromones. And I understand Jeff Aldrich, who's the bug specialist on pheromones in the world, is now in your department, and he can fill you in on all that background with that's a lot of similarities between bed bugs and many other bugs that use alarm pheromones. There's some evidence that, and good evidence, that there's a pheromone that mediates their aggregation at those hidden cracks and crevices. There's also evidence that these pheromones are involved in sexual and stage differentiation. So what this means is that a male bedbug will jump on anything that moves in its environment, but the individual that it jumps on will release an alarm pheromone that indicates it's a wrong stage or that it's another male, and that discourages the attempt. So those are recent publications dealing with how those quote-unquote alarm pheromones work in bedbugs. One thing that I've been really interested in is, with another graduate student who's working with me, is whether there's any evidence of a relationship between mother and offspring that might be mediated by pheromones. And I just go through a few of the evidence of pheromones involved in bedbugs. Here's a little demonstration. This actually followed the original protocol of 1974, demonstrating some aggregation stimulated by pheromones, where we released a group of bedbugs at the center, and they re-aggregated on paper marked with fecal material from bedbugs. And if you extract the feces of bedbugs, that will stimulate with a solvent like methanol. That extract will stimulate them to re-aggregate. It's a very convincing demonstration to see them re-aggregating based on a chemical stimulus alone. Now, our interest in whether bedbugs might interact with their offspring is just simply coming from the fact that moms and offspring are together in the same place for extended periods of time. And that's shown here in this example. Also shows the size difference between an adult female and first instar nymph. There are reports of these mother-offspring pheromones in a number of other insects, including the burrower bug that I worked on pheromone identification and with a colleague over in Switzerland. And he demonstrated very effectively that the nymphs beg for food with a chemical signal, and that mom brings the food to her early instar nymphs. The food is a mint seed in this case. And then the other slide is of an earwig that also has a chemical signal involved in the interaction between mom, who's hovering over the early instar earwig nymphs. This little video shows a female bedbug laying an egg, You can see there's already a fecal spot in this area, and behind that fecal spot, there's an egg. And she's getting ready to lay another egg. It's certainly involved operation here. We produce the egg, but this is what I'm interested in. After she lays it, this is all happens every 100% of the time. She does that little maneuver over the egg. And I'll show you another example here. She's just laid an egg, the female at the top of the video, and she's spending quite a bit of time on this effort. So what I've suggested, and this is work that is still to be done, that we hope to do soon, but I'm suggesting that she's marking the egg. It could also be that she's putting down antifungal materials or something like that with the egg. So there are other hypotheses as well, but of course I'm interested in the aspect of chemical communication. I did want to introduce you to some work that's been done by Sidney Crawley, a graduate student, on this topic. This has nothing to do with that marking of the eggs, but it has to do with their feeding behavior. And how we feed bed bugs is, rather than strapping them to our legs, we use a feeder. Bob, you've probably had experience with these types of mosquitoes, with feeders with mosquitoes and so forth. This is one that's fairly commonly used with mosquitoes and other blood-feeding insects. But a parafilm membrane is stretched over the bottom part of this cylinder that circulates heated water, and the blood that's pooled at the bottom of that is available through the parafilm membrane to bed bugs that climb up through this cylinder to get to it. And so this is we use heated rabbit blood as a source of food for these bed bugs. And that just shows photographs of the same apparatus. And what Sidney Crawley was able to show was that in the presence of females, the first instar nymphs were much more effective in finding that food than they were in the absence of the adult females. Males and females together would stimulate the increase that was shown with females only. But when males were with the nymphs alone, they did not improve the feeding success of those first instar nymphs. So the increase that we saw of first instar feeding was always associated with the presence of females. So we did another study in this system where we had a digital camera situated below the cylinder so we could view the feeding activity over time, recording the picture every 30 seconds. And this is what one of those pictures looks like when it's taken in the dark with the infrared illumination so we can see what's going on. And you see 3 fully engorged females there, and there are other fully engorged females in the picture that we've recorded over time. They're moving up to feed on the membrane. So this is a view of the top of that cylinder as they feed. There's a first instar nymph that's moved up to that surface and is starting to feed. And what we found with this recording was that the adult females arrive on average before the first instars, which of course would be critical if there's signaling going on. Females would have to get there first in order to signal that they've discovered a food source. And that's what we find with this time course, that the adult females get there first, And on the average, it's about 5 minutes before, 5-6 minutes before the nymphs would get there. So the timing is right for there to be a chemical signal involved. Here we did an experiment where the initial treatment was to feed females, and we had this paper bridge that they could walk up to the food, up the cylinder to the food resource. So we pre-exposed filter paper bridges with females that went up and fed. Then we moved the filter paper over to another cylinder where we placed first instars. You can barely see them because they're so small. And the same with filter papers that had no exposure. And if there's a chemical signal that endures on the filter paper, we'd expect to see a difference in these two treatments. That's just to give you a little background. You can say you've seen everything now. So one possibility that it's also from fecal spots that are left on the filter paper. And if we look at the top end of the filter paper, which would be closest to the food that they're engorging on, we do see fecal spots. There are actually at least two types of fecal spots, the dark ones, and then there are ones you can barely see unless you use UV light to look for them. UV light, we thought when we found this, the UV light really makes the feces shine in the dark. We thought we were onto something. But then the mattresses all shine in the dark too when they're under UV light, so it didn't work out for us. But there was an improvement in the efficiency of first instars finding food when that bridge treated with the females themselves feeding was put there. So it could be from the fecal spots, or it could be from some other pheromone contamination of the filter paper. But this is another research area that we're trying to track down right now. And one of our approaches has been to do volatile collections from adult females and adult males as they feed. And we definitely associate compounds with feeding. You can look here in the female, materials that they're releasing while they feed. And these are the same alarm pheromones that were discovered in 1974 and are usually not associated, have never been associated with feeding per se before, always been associated with disturbance of bedbug colonies. But the females release a different ratio of these compounds than the males do. You'll see a tiny amount in the male there, and in this male, a different ratio than the female. So it opens the possibility that there's a pheromone signal that's released by females during feeding, that it's attractive to the nymphs. And just another hypothesis we want to explore in great detail soon, because I think this could be really, really critical, something that's been missed before that we want to nail down here. So it's an active research project right now. Now, I just want to close up and summarize here, but with this slide. Back to this slide. This is a photograph taken by my long-term colleague at Kentucky, Mike Potter, who's an extension entomologist, but was the one who convinced me to bring in this student, Alvaro Romero, who got me started on the bedbug research. My background was all on moths, so this was a new venture for me, and a lot's going on in this harborage. There's some unusual mating going on. There's a deposit. Not all that many animals deposit their feces in the same place that they reside for 90% of or more of their life. Eggs are in the same environment, all stages occurring in this small place. Somehow they have to get out to a host, discover the food, and we know that that's mediated in part by a circadian rhythm of activity, but it also is influenced by heat and probably a different way by CO2. And their pheromone involved in aggregating in spots like these, but also moving out to finding their food resources. That the pheromone signal that seems to be involved with the female communicating to nymphs, the males actually listen in on that conversation, and they go to the food source too to a greater extent when there are females present than they would on their own. So that's a good place to find food and other things. I would like to acknowledge Tom and Nina Lee for funding the endowment that's allowing these kind of seminars to take place, and the Department of Entomology and Nematology here for inviting me. It's been a great experience. I would also like to acknowledge this group of people. This is Mike Potter, Sidney Crowley, Jennifer Gordon, who's worked on resistance to pyrethroids and neonicotinoids, Mark Goodman, who's working on symbiosis of bed bugs with bacteria, which I think is really interesting work. Technical support from Scott Besson, and some funding opportunities that we've pieced together to support our bedbug research program. Some of the work that was done on insecticide resistance was spearheaded at the physiological level by Reddy Pauly and Fang Xu. And Alvaro Romero, I have to give a special thanks to for introducing me to bed bugs, although Mark Feldhofer and Mark Epper were the first that introduced me. He's the first one to get me excited about doing research on bed bugs. So I thank him as well. And thank you all very much for coming.
Any questions? Time for questions? You want to ask a few questions?
Yes.
You said one of your students found 3 mattresses out of 3 with bed bugs and no indications. I took indications to mean the salt and pepper sign, the feces in it.
Oh, no.
Okay.
She found those kind of indications. What I meant is there's no human indications.
Oh, okay.
What they're supposed to do when they throw a mattress out is to mark it extensively with markers or paint and indicate bed bug infested, so that it doesn't travel to another apartment in the next day. So I wasn't clear on that. I'm sorry.
No, thank you.
Yes.
So one of the big fears that we have when we go to hotels is that they invade our suitcase and our luggage, but it doesn't seem like our luggage would have that much heat or carbon dioxide. So are there other chemicals that attract them, or do they just stay in our jammies after they drink our blood?
There are reports that body odor plays a role in their finding host, but on top of the other things, it must be a relatively small role. But I would say that their tendency to move, even in the absence of hosts, is probably more important way they would get into your suitcase. Because that one bed bug that my student Becca Hassel tracked for only 5 minutes, traveled 4 meters. It wasn't all in a straight line towards your suitcase, but if you multiply that by moving around throughout the night, they could cover a lot of territory. And so they're random movements, and how many female bed bugs invading your suitcase does it take to be a problem? I think the measure that I take when I go to a hotel room is to inspect the bed. If I find no evidence around the cracks and crevices of the box spring or mattress of bed bugs, then I feel pretty good about the population being low enough that I won't take one home with me. And I don't mind a bite or two as long as I don't take one home, because my wife says that I have to find a different research topic if I start bringing it home.
Okay, we have two more questions, please.
So why do you think they don't vector? I mean, so you go back 20 million years and all this time they don't vector any. And also, how many other groups are there where blood feeding and they also do not vector a disease?
That's a very good question. I think there are certain biological characteristics that would make them not a very good vector. They typically would be feeding on the same host once a week. So they're staying near the host bed. So the potential to transmit something to a new individual is much less than an insect that has greater mobility that can fly.
But lice and fleas
Yeah,
are in that situation too.
well, I'm not sure that they're quite as much as a homebody as a bed bug. But the bed bugs will fully engorge every time they feed, and then they will not feed again typically for a week or more. So there's also a timing thing there. How persistent would the disease organism have to be in the bed bug for it to be a good vector? But there are things like that that might make the biology less well adapted for transmitting a disease. But I don't know the answer why there is not an association. I'm not sure how many people have looked at a disease like Chagas disease to see if could a bed bug vector Chagas disease? I don't know. It's some of the things you don't explore. We did look at some transmission of bacterial organisms, and we found that it would pass through the bed bug. This was Borrelia species, and it would pass through the gut and would be deposited in the feces, but it would not persist in the environment. So I think it's part biology and part we don't know yet. Yes?
Is there any indication that the pheromone produced by the mother would influence her own offspring more than some other mother's offspring?
That is, of course, a very good question, and we don't know the answer to that. We try to use, in these experiments, mothers with their own offspring, and we did that for several goes around, and we found the same type of results that I presented here. So there doesn't seem to be a mother-specific pheromone involved, but there's certainly going to be an association between mother and her offspring that potentially could make it work. Yes?
Sorry. Can you tell me a little more about traumatic insemination? Why bother?
Why circumvent the normal mating for the alternative? I mean, it's a very good question. The story in the literature is that there's a conflict of interest between what males are out of in reproduction and what females are out for in reproduction. And this traumatic insemination started with males circumventing the female choice in the process. And that a secondary effect is the female adaptation for that traumatic insemination event. If you look at the whole family of semicity, you find some in which the females have this adaptation to receive puncture wounds, and other females other species that don't have it. And there are also males that have the female, the same adaptation that the females have to apparently encourage traumatic insemination from males to compete, to take mating opportunities away from other males.
Okay, when that happens, so does the male, I thought the male could actually pass sperm that wasn't his own. Is that incorrect?
No, he's passing his own sperm, and his own sperm takes precedence in mating with a female. Is that, what was the context that you?
So I thought that if a male mated with another male, and the other male mated, he'd pass some of the original, the original male's sperm was out.
I mean, if that's reported out there, it seems very unlikely to me because of the pathway that it would have to go through to get into the male reproductive tract and then get into the accessory gland fluids and then move on with the ejaculate. So you never know. I've been surprised by bed bugs many times.
Okay, going to bring it to a close. I think probably all of us will pay more attention when we visit some of these hotels, when we look for a mattress and see what's going on there. So that's a positive side of this presentation. I do want to mention that last year, I think 2012, Ken published an article in Scientific American. The title was, I think, Sleeping with the Enemy. So if you want to go back and review that, it was a review article. He was invited to do that, and you can see why. Obviously, general information for sort of the lay public, but then laced with good science as well, and that's what we heard here. So 3 things I want to do is make three presentations, okay? The first is we're going to memorialize this event by putting Ken's name on a plaque that will be in the hallway outside of the Department of Entomology main office. That's the first. Okay, the second is that we do have a very nice plaque for Ken that says presented for the Thomas and Nina Lee Distinguished Alumni Seminar to Ken Haynes, November 20th, 2013. Give that to you. Okay, and then we also, again, Tom and Nina Lee have sponsored this event. We do have a $500 check to give Ken as an honorarium for this presentation. And again, I want to thank everyone for coming. It's a wonderful affair, and there's no question that Ken is an outstanding alumnus, and this has been a tremendous success. So let's thank Ken, and thank you for coming. One more thing. To put this together takes a little time and effort. We did have 2 staff. Obviously, Lisa Gerardo didn't stay. I don't think she's not here, but Amanda is sliding back, hiding. And Janet, obviously, to put this together, the brochures, the plaques, and things like that, takes a little bit of an effort. We do have a tremendous staff here in entomology, so let's acknowledge them as well. Thank you.